Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi ta'ala wabarakatuh. Hello everyone, my name is Nazrina and welcome to our first lecture for UMC 463 Meet Your Microbes, entitled Bacteria, Characteristics, Classification and Importance. By the end of today's lecture, it is hoped that you should be able to do the followings. The first, hopefully you should be able to describe the basic characteristics and classifications of bacteria. And secondly, hopefully you should be able to demonstrate your understanding of the beneficial roles and clinical significance of bacteria. Let's start first with bacterial characteristics. The term bacteria actually means small stuff, and the term was first used back in 1838. Bacteria are essentially microscopic, unicellular, prokaryotic organisms, which means that they have no nucleus and they have no complex organelles. They're simple stuff, but do not underestimate these little microorganisms. Bacteria come in many roles. They wear different hats. Some are pathogens, which means that they're always up to no good. They cause and they spread infections. Second are non-pathogens. They're usually organisms that are harmless by nature. They're just living their life and they're minding their own business. And lastly, there are the commensals. They're usually friendly organisms, but they can become opportunistic when your immune system becomes impaired. So what's inside a microbe? Say you have an organism and you dissect it. These are the things that you may or may not find inside a bacterium. The first is, as I mentioned before, they lack a nucleus and they lack complex organelles. Secondly, they possess cell membrane and they possess a cell wall that is made out of peptidoglycan. The thickness of the peptidoglycan is very, very important for the purpose of classification. I will talk about it later on. They may or may not possess flagella or pili. Flagella is very important for movement and interaction. They possess plasmids, which are essentially small circular DNA. Some may have capsules, and if they have capsules, it is advantageous for the bacteria because the capsule's role is actually to protect the bacteria from phagocytes. They have adhesins, which is for binding and attachment purposes, and they will release enzymes and toxins, which are essentially their weapons or the virulence factors which they use when they want to infect a particular host. Bacteria are tiny organisms because the term means small stuff, remember? But how tiny is tiny? Well, essentially, bacteria size ranges from 0.2 to 5 micrometers in length. So if you could put things in perspective, say you have a salt grain, one salt grain, you put it on your finger. A bacteria is 1,000 times smaller than that one particular salt grain. So that's how tiny it is. But obviously, if you want to compare against viruses and fungi, they're much bigger than the viruses, but they're much more smaller than the fungi. So now let's move on to bacterial classifications. Bacteria, like all living organisms, are categorized, grouped, and classified into a hierarchical ranking system, which we call bacterial taxonomy. We start first with the domain, which is the highest ranking group for the classification system. Then we've got the phylum, which groups bacteria based on major genetic and structural traits. You've got class, which is a subdivision of the phylum. You've got order, which groups related families. You've got the family, which groups related genera. You've got genus, which is a group of closely related species. And lastly, we've got the species, which is the most specific level in the classification system. Other than bacterial taxonomy, you can also classify bacteria based on a number of factors, such as based on their shapes and arrangement, based on their gram stain, based on their flagella, and based on their nutrition, oxygen, and temperature requirements. So let's start first with classification of bacteria based on their shapes and arrangements. So some bacteria are considered coccus or cocci because their shape is spherical or oval in nature, examples of which include Staphylococcus and Streptococcus species. Then there's another group of organisms which we call bacilli, and these group of organisms, their shape is much more elongated or cylindrical, and examples of which include Bacillus, E. coli, Klebsiella, Enterobacter, and Citrobacter. And lastly, there's a group of organisms which we call spiral, and this is further classified into either curved or helical bacteria, and examples of which include your Vibrio, Spirillum, and Spirochetes organisms. Now let's have a closer look at cocci, your spherical, round, oval organisms. Cocci can be further classified into either being monococci, diplococci, staphylococci, streptococci, tetrads, or Sarciniae. So what's the difference? They are all the same in terms of their shape, they're all oval, but the difference lies in the way they are arranged. Monococci, for example. Mono, as the name suggests, means one, which means that they are essentially singular cocci. For example, are Micrococcus luteus. Then you've got your diplococci, which are essentially two bacteria arranged in pairs, example of which include your Neisseria species. Then you've got your streptococci, which are cocci, spherical organisms, but they are arranged in chains, just like arrangement of a necklace. Examples include Streptococcus pyogenes. Then you've got staphylococci, which are cocci arranged in clusters. So if you can imagine, arranged like grapes. Here, examples include your Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus, all arranged in a group of clusters. Then you got tetrads, which are Staphylococcus arranged in a group of 4 cocci, such as your Aerococcus species, and Sarciniae, which are your cocci arranged in a group of 8. Example are Sarcinia species. Now let's move on to our bacilli, which are essentially our rod, elongated, and cylindrical organisms. Just like our cocci just now, the shape of the bacilli could be the same, but the arrangements could be different. For this reason, bacilli can be grouped as the following. They can be bacillus, monobacillus. They could be diplobacilli, streptobacilli, palisade, and coccobacilli. So monobacillus is essentially one single bacilli. Example of this is Salmonella typhi. You've got Moraxocella bovis, which is essentially a diplobacilli because they are bacilli that are arranged in a pair. Then we've got a group of organisms called Streptobacillus monuliform, which are your streptobacilli, which means that your bacilli are arranged in a chain. Then we've got Corynebacterium diphtheriae, which causes diphtheria, which are essentially palisades because the arrangement of the bacilli is in the form of a fence. And lastly, we've got coccobacilli, such as Chlamydia trachomatis, which are essentially bacilli, but they have a rounded end. So other than your cocci and bacilli, we've got organisms that are either spiral or comma in nature. For spiral, these are long helical shaped or twisted bacteria, examples of which are your spirochetes. And comma, there are essentially comma or curve-like structures, for example, your Vibrio species. Not all bacteria can be neatly classified as being either cocci, bacilli, helical, or comma shaped. Some are actually more miscellaneous in nature, and this includes our pleomorphic organisms such as our Mycoplasma, our bacteria that are arranged in Chinese letter arrangement such as Corynebacterium, our filamentous bacteria such as Actinobacteria, and our star-shaped bacteria such as Stella vaculata. One of the most fundamental ways to separate or classify bacteria is via gram stain, whereby this procedure allows us to separate organisms based on their peptidoglycan layer, So organisms are either gram-positive or gram-negative. A gram-positive organism is a bacteria that has a thick peptidoglycan layer which enables it to retain the purple color of the crystal violet stain that is used during the gram staining procedure. Examples of gram-positive organisms include Staphylococcus, Streptococcus, Enterococcus, Corynebacterium, Bacillus, Clostridium, and Listeria. In contrast, gram-negative organisms have a much more thinner peptidoglycan layer, and for this reason, it cannot retain the purple color of the crystal violet, and instead, it retains the pinkish red color of safranin. Examples of gram-negative organisms include E. coli, Klebsiella, Enterobacter, Citrobacter, Salmonella, Shigella, Serratia, Proteus, and Pseudomonas. For this reason, gram stain is a fantastic way to classify bacteria based on the color of their stain retention when seen under the microscope. So bacteria are classified either being gram-positive, which will appear purple under the microscope, or gram-negative, which will appear pinkish red under the microscope. So examples of gram-positive cocci include Staphylococcus aureus and Streptococcus pyogenes, and gram-positive bacilli include Bacillus anthracis and Listeria monocytogenes. In the gram-negative or pink team, we have gram-negative cocci such as Neisseria gonorrhea, Moraxosella catarrhalis. You've got your bacilli, you've got E. coli, Klebsiella, Enterobacter, Citrobacter, Salmonella, Shigella, Neisseria, Pseudomonas. And you've got your comma-shaped gram-negative bacteria, such as your Vibrio species. Flagella, as I mentioned before, is very important for movement and locomotion. Its existence, as well as its arrangement, can be used to classify bacteria. Because there are certain bacteria that doesn't have any flagella. Some may have single, some will have many, and some may have all over the place. Let's start first with Atrichus. Atrichus means bacteria that doesn't have any flagella, is non-motile. Example of this is Bacillus anthracis. Secondly, Monotrichus. Mono means one. This particular bacteria possess a single flagella that is situated at one pole. Example of this is Vibrio cholerae, whereby it only has one single flagella, but it's a very powerful flagella that is responsible for its darting motility, as observed for Vibrio. Then you got Lopotrichus. Lopotrichus means bacteria has a cluster of flagella, not single, but a cluster that is situated at one end. Example of this is Helicobacter pylori, the causative organism for your gastric ulcer. Then you got Emphatrichus, which means bacteria has flagellas at two opposite ends, not one but two. Example of this is Alcaligens faecalis. And lastly, you got your Peritrichus flagella, which means the bacteria is covered with flagella all over the surface, like rambutan. Example of this is E. coli. Next is classification based on oxygen requirement, because oxygen requirement is very, very unique. Some bacteria requires oxygen, some can die in the presence of oxygen, some requires very little oxygen, some don't mind if they have little or if they don't have any oxygen. So this is the rationale of the classification based on oxygen requirements. Obligate aerobes are bacteria that requires oxygen. It's a must to have oxygen. Without oxygen, they will die. Example of this is Pseudomonas aeruginosa. Anaerobic bacteria, in contrast, are bacteria that cannot survive in the presence of oxygen. You give them oxygen, they will die. If they don't have oxygen, they will be happy. Example of this is Clostridium botulinum. Then you have a group of organisms called facultative anaerobe, which means they're very, very flexible. They can grow in the presence or in the absence of oxygen. They're really okay with either way. Example of this is E. coli. And you got microaerophiles. Microaerophiles means that they require oxygen to live, but they just need a small amount. These are microaerophiles, example of which are Helicobacter pylori. Next is bacterial classification based on temperature requirements. Some bacteria can grow wonderfully at room temperature. Some can withstand very cold temperature. Some can withstand very extreme hot temperature. And that is the basis of the classifications. So psychrophiles are bacteria which can grow optimally at 15 degrees and below. For example, Listeria monocytogenes can withstand a very cold temperature. We've got the mesophiles, which are bacteria with an optimum growth temperature at between 15 to 45 degrees Celsius, your normal bacteria such as Staphylococcus aureus. And you've got thermophiles. These are very, very unique bacteria which can grow optimally at temperatures exceeding 45 degrees. You imagine there are certain bacteria that can live happily in hot springs. For example, Geobacillus thermocarbonatus can survive at hot springs. Similarly, Bacillus thermophilus can also survive very, very high temperature, and these are examples of thermophiles. Another way to classify bacteria is by how they obtain their nutrition. Bacteria could be autotrophic, heterotrophic, or saprophytic. Autotrophic means that bacteria can actually make their own food. Heterotrophic means that bacteria obtain nutrients from others. And saprophytic means that bacteria feed on dead, decaying organic matter. Autotrophs are bacteria capable of assimilating inorganic matters into organic matters, which essentially means that this particular organism can make their own food. There are two types of autotrophs, which are photoautotroph and chemoautotroph. So what's the difference? Photoautotrophs are bacteria that uses energy from sunlight for assimilation purposes, examples of which are cyanobacteria. In contrast, chemoautotroph uses chemical energy, not sunlight, but energy from chemicals for assimilation, and these include sulfur bacteria and nitrogen bacteria. Heterotrophic bacteria cannot make their own food like autotrophs. Instead, they derive energy by consuming organic matters from others. In this respect, this group of bacteria can be seen as parasitic or symbiotic in nature. Examples of heterotrophic bacteria include E. coli and Staphylococcus aureus. And lastly, there's a group of organisms known as saprophytic bacteria, and these are bacteria that decompose organic compounds, and from there they get their energy. So essentially they are decomposers, and they feed on dead and decaying matters either from plants or from animals to get their food, examples of which include Pseudomonas aeruginosa. Now let's have a quick recap for the first course learning objectives. Hopefully by now, number one, you must have appreciated the fact that bacteria are tiny, microscopic, unicellular prokaryotes that are ubiquitous in nature. They're found everywhere. Secondly, bacteria are classified in many ways. You can classify them taxonomically, or by their shape, by their gram stain, by their flagellar arrangement, by the oxygen requirement, temperature requirement, or by how they obtain their nutrition. Thirdly, gram stain is a fantastic procedure that can easily classify bacteria in two main groups, either a gram-positive organism or a gram-negative organism. A gram-positive organism is an organism that possesses a thick peptidoglycan layer, and upon staining, what happens is they can retain the purple color of the crystal violet, and for that reason, it appears purple under the microscope. A gram-negative organism has a thin peptidoglycan layer. It cannot retain crystal violet, so instead it retains the pink color of the safranin, and for that reason it appears pinkish red under the microscope. And hopefully, lastly, by now you must realize that there are many shapes of bacteria, cocci, bacilli, spiral, comma, and hopefully you'll be able to give examples of each one. So that's it for basic bacterial characteristics and classifications. I hope you're ready to meet the good guys and the bad guys of the bacterial world. So now let us look at the beneficial roles of bacteria. Let's take a step back and look at the positive sides of bacteria, how they can become heroes in disguise, how bacteria can help mankind. Ladies and gentlemen, boys and girls, there is a whole list, a quite extensive list as to why we should love our bacteria. Because bacteria actually does a lot of good in our daily living. We may not actually realize it, but they're actually good guys. They can recycle elements such as nitrogen and carbon to keep life going. Some organisms such as cyanobacteria can actually make oxygen. A lot of bacteria are actually very active in cleaning up the environment. They do a lot of CSR. They are involved with biodegradation, composting, and bioremediation. Bioremediation is a mouthful. It's very difficult for me to even pronounce it, but essentially it's a process whereby you use organisms to break down, to neutralize, or to remove pollutants. For example, you've got oil spillage in the ocean, you can actually use organisms to clean it up. So it's wonderful for the ecosystem. There are fantastic industrial helpers. They make enzymes for food, for medicine. They are bioengineering buddies. They're very, very useful in the pharmaceutical industry to produce things like insulins, hormones, genetically modified organisms. They're gas producers. They make biogas such as methane for fuel. They're antibiotic heroes. They save life. I know a lot of bacteria will be killed by antibiotics, and bacteria also are the organisms to actually produce antibiotics as well. They are gut guardians. Probiotics is good for gut health, and for that reason you can find lots of drinks, for example vitagens, that contains healthy, loving, friendly bacteria. Tempoyak is another food delicacy that contains a lot of probiotics. They contain a lot of lactic acid bacteria that is very good for your gut and good for your well-being as well. They're culinary masters. They're used for fermented goods. They're used to ferment cheese, to ferment yogurts. They are farm friends. They are used for biofertilizers and biopesticides. They are soil boosters, and they are ancient pioneers. They make Earth habitable. They have been here forever, and they make Earth the lovely place that we live in right now. So yes, there's a lot of good things about bacteria, but obviously there's a lot of bad things about them as well. So now let's dive into the clinical significance when bacteria go bad. So bacteria that causes infections or diseases are known as pathogenic bacteria. And pathogenic bacteria, they cause infections not only in humans, but they cause infections in humans, animals, and also in plants. And some, although initially they do not cause harm, when your immune system is weakened, when it's hampered, or when it's impaired, it can cause opportunistic infections. And these are what we call opportunistic pathogens. Okay, when we talk about clinical significance, if I was to list down all the infections caused by bacteria, the list can go on and on and on and on till Jannah. So here I'm just going to summarize some of the most common bacterial infections and some of the organisms that actually is associated with them. Examples of which include Staphylococcus aureus, which can cause skin infection such as Staphylococcus scalded skin syndrome, which is actually a dermatological condition caused by Staph aureus. Staph aureus is also associated with food poisoning, UTI, toxic shock syndromes. Streptococcus pyogenes that causes strep throat, scarlet fever, rheumatic fever. E. coli. E. coli is a champion, is associated with a lot of things including UTI, gastrointestinal tract infections, neonatal meningitis. Salmonella is associated with typhoid fever, paratyphoid fever, salmonellosis. Neisseria meningitidis, as the name implies, is associated with meningitis, septicemia. And then we have Mycobacterium tuberculosis, which is the causative organism for tuberculosis, as the name implies. Usually it's abbreviated as TB, which is a respiratory tract infection that has been around since ancient times. We've got Vibrio cholerae that causes cholera, Clostridium tetani that causes tetanus, Clostridium botulinum, which causes botulism, Helicobacter pylori, which is associated with gastric ulcer. So besides infections and diseases caused by bacterial pathogens, Bacteria can also cause a wide range of negative impacts to the environment and to several industries. Bacteria can cause spoilage in food and medicine, causing economic loss. They can reduce soil fertility, resulting in smaller crop yields. Bacteria can damage crops and damage livestock, which is bad news for farmers and entrepreneurs because it will result in lower food supply. And they can destroy goods such as furniture and textiles, again causing economic loss. So now let's recap. Bacteria recycle nutrients, they can make oxygen, they do wonders for our pharmaceutical industries. They are the sources of antibiotics, the sources of enzymes, and the sources of fermented foods, which brings a lot of advantages to mankind. A lot cause diseases, obviously. They can spoil food, they can damage crop, they can harm the environment. So in essence, it is wise to summarize that some are beneficial, some are also harmful. So it's our task to understand them and to use this knowledge wisely because balance is key. So this is the take-home message. Love and appreciate bacteria, our tiny neighbours who are small, mighty, but very, very significant. That's all for now for Bacteria 101. Thank you for joining us. Should you have any questions or should you want to know more about bacteria, please do not hesitate to contact any one of us. All the 5 instructors are ready to help you in any way we can. Thank you very much. Assalamualaikum.